All hail the king of the termites. The bugs are unique among colony-forming insects in that their queens cannot rule without a king. While millions of eyes are on the coronation of Charles II this weekend, other male monarchs will carry on ruling in obscurity, literal obscurity, because they live in darkness underground, or maybe in a piece of rotting wood. Queens are familiar figures among insect species, but it is only in termites that royal families include kings, long-lived, ensconced in fortified royal chambers and faithful to their enormous queens. These rulers are unique in the insect realm. There are some parallels to our monarchy system for humans, said Barbara Thorne, a termite biologist and professor emerita at the University of Maryland. Termites, like many ant, bee and wasp species, live in collaboratives of sterile workers. But there are crucial differences. In the nests of ants, bees or wasps, the sole monarch is a queen. She stores all the sperm she will need for the rest of her life after a single, eventful mating flight. She uses the stored sperm to produce daughters, who take on all the work of finding food and caring for their younger siblings. Termites evolved separately from these other insects, and they do things their own way. For starters, both a queen and a king are needed to start a termite colony. The reason isn't romantic. Female termites can't store sperm, so they need a male to inseminate them over and over. A study of one species found that it happened daily. King and queen work together to get their family started. The male helps with every aspect of it, Dr. Thorne said, from the brood care to nurturing, feeding, providing water, attending his queen. Soon, though, the royal couple steps back. They keep making babies but let grown offspring handle the forage and child care. Those helpful kids are both male and female, a society of brothers and sisters working together. Some also grow up to become soldiers specially built for defending their colonies. The queen and king don't set themselves apart with crowns or other regalia, but they do start out in adulthood with wings, which they shed as they're settling down. The queen becomes ginormous, just a big sack of ovaries, basically, said Ed Vargo, an entomologist at Texas A&M University. The king, while dwarfed by his bride, is still larger than the workers and usually the soldiers. Termites can't see those differences as they live in the dark, but Dr. Vargo's lab has shown that workers and soldiers can recognize their sovereigns by smell. When another termite is near a queen or a king, it shakes its body, perhaps alerting others that they're in the presence of royalty. And although they don't have a castle, Dr. Thorne said, they do have a special royal cell with a little chamber that's primarily the queen and king. Deep within the nest and with thicker walls, this chamber protects the queen and the king if, say, an anteater takes a swipe. As the royals don't leave their chamber, workers give them regurgitated food from their own mouths. Other colony-living insects spike their mouth-to-mouth meals with chemicals for communication, Dr. Vargo said. It's possible that termites do the same thing. For now, though, the makeup of these termite meals is as mysterious as the secret mixture of oils with which Charles II will be anointed, using the centuries-old coronation spoon. The termite king, performing his marital duties, helps his mate produce as many as 15,000 eggs each day. When the king or the queen eventually dies, a replacement will come from among these heirs and many, many spares. The reproductive throne is inherited by one or more of their offspring, Dr. Thorne said. In rags to riches style, the first termites evolved from cockroaches. Why they became so cooperative is a bit of a puzzle. Ants, bees and wasps have a genetic system that makes the females in a family more closely related than usual. This may be part of why queen-led colonies evolved several times in these insects, but a termite colony is no more related than a human family, at least until the inbreeding starts. Whatever led them to evolve cooperative societies, Dr. Vargo said, it happened only once in history. Today they have expanded into about 2,600 highly cooperative species. Termites are amazing, and super important in their native habitats for decomposition and recycling of nutrients, Dr. Thorne said. But our interactions with those societies are mostly negative since, she said, we build our houses out of termite food. The termite monarchy also offers a window into the science of longevity. Like ant, bee or wasp queens, termite royals can live substantially longer than their subjects. Workers may live a few months, while termite royals have survived 20 years in laboratories. Isaac Tasaki, a biologist at Niigata University in Japan, said termite kings have a distinctive set of molecular tools that slow their aging, such as more active genes for DNA repair. Unraveling the molecular mechanisms of termite king longevity may shed light on the mysteries of lifespan regulation in male individuals in all animals. Dr. Tasaki said, "Long live the king, indeed. Aging is associated with the accumulation of DNA damage. High expression of DNA repair genes has been suggested to contribute to prolonged lifespan in several organisms. However, the crucial DNA repair genes contributing to longevity remain unknown." Termite kings have an extraordinary long lifespan compared with that of non-reproductive individuals such as workers, despite being derived from the same genome, thus providing a singular model for identifying longevity-related genes. In this study, we demonstrated that termite kings express higher levels of the breast cancer susceptibility gene BRCA1 than other casts. Using RNA sequencing, we identified 21 king-specific genes among 127 newly annotated DNA repair genes in the termite reticuloderm spiratus. Using quantitative PCR, we revealed that some of the highly expressed king-specific genes were significantly upregulated in reproductive tissue, testis, compared to their expression in somatic tissue, fat body. Notably, BRCA1 gene expression in the fat body was more than fourfold higher in kings than in workers. These results suggest that BRCA1 partly contributes to DNA repair in somatic and reproductive tissues in termite kings. These findings provide important insights into the linkage between BRCA1 gene expression and the extraordinary lifespan of termite kings. Genomic instability and DNA damage are hallmarks of aging and number of aging-linked diseases in multicellular organisms. Indeed, the frequency of DNA damage increases with age in humans, rodents, and flies. Aging-associated DNA damage is normally repaired by various DNA repair proteins. Thus, these proteins are highly conserved in many organisms and they play important roles in organismal aging and longevity. In mammals, longer-lived species such as humans and naked mole rats exhibit higher expression of DNA repair genes than short-lived mice. Moreover, overexpression of several DNA repair genes contributes to the extension of lifespan in the fly Drosophila melanogaster. These studies suggest that efficient DNA repair system contributes to extended longevity. However, the crucial DNA repair genes contributing to extraordinary longevity are unclear. In eusocial insects, such as ants, honeybees, and termites, the lifespan of queens is more than tenfold longer than that of non-reproductive individuals, workers and soldiers, possessing the same genome, a fact that has attracted significant attention. In contradistinction to the queens, reproductive male ants and honeybees have a relatively short lifespan, 2-3 months. Conversely, termite kings exhibit extraordinary longevity similarly as the queens. This illustrates that termite kings have an exceptional anti-aging system. A previous study focusing on somatic tissues reported that long-lived queens displayed higher expression of DNA repair genes than short-lived workers in the ant Lasius niger. These findings suggest that highly efficient DNA repair mechanisms can also contribute to the extraordinary longevity of termite kings. The Japanese subterranean termite Reticuloderm spiratus is one of the most studied termites due to its reproductive system. A novel parthenogenetic system known as asexual queen succession (AX) was revealed in R. spiratus. AX allows queens to extend their reproductive lifespans because a founding queen, primary queen, PQ, is genetically immortal until a colony dies due to the production of a large number of nest queens, secondary queens, SQS, through parthenogenesis. Conversely, a founding king, primary king, PK, should continue to live even if queens are replaced because mother-son inbreeding depression can occur when the PK dies, reducing the genetic contribution to offspring. Indeed, a previous study has revealed that this king replacement does not often occur in wild colonies of R. spiratus. It is thus presumed that PKS in R. spiratus have been exposed to selection for inbreeding avoidance. As a previous study reported that Reticuliformes termite queens surprisingly live more than 11 years, the PKS of R. spiratus may have the potential to live for several decades. Therefore, R. spiratus provides an ideal model for identifying the critical DNA repair genes contributing to extended longevity. In this study, we performed transcriptome analysis of DNA repair genes using RNA sequencing (RNA-seq) data obtained from all casts of R. spiratus, both females and males, workers, soldiers, elates, young PQS and PKS, and mature SQS and PKS. We additionally investigated age-dependent expression changes in male reproductives, male elates, and young and mature PKS. Furthermore, we investigated where DNA repair genes were overexpressed in the termite body using quantitative PCR, qPCR, analysis.